Good afternoon, everyone. Welcome to the Association for Molecular Pathology's webcast in the Emerging and Evolving Biomarker series on methylation profiling. Before I turn the floor over to our moderator today, I have a few housekeeping items to discuss. This session is being recorded and all attendees have been placed in a listen-only mode. If you have trouble hearing the presentation via your computer speakers, please select the phone call option under Audio on your GoToWebinar control panel. Information on how to dial in via your phone will populate for you. You are also able to change your speakers by selecting this drop-down. All audience questions will be addressed at the end of the presentation during our Q&A session. However, you are highly encouraged to submit your questions at any time throughout today's webinar. Please type your questions into this panel and click Send. Thank you for joining us for today's live webinar, and I will go ahead and turn the floor over to our moderator, Dr. Anthony Snow.
Good afternoon, I'm Anthony Snow. I'm a molecular genetic and GI pathologist joining you from the University of Iowa Hospitals and Clinics today. I'm a member of the content committee for the AMP Emerging and Evolving Biomarkers series, and I'll be your moderator today. Thank you for attending today's webinar on methylation profiling. Before we begin our webcast, I'm going to ask you 6 questions as a pre-quiz. This is designed to test your prior knowledge of the topic, so don't worry if you don't know the correct answer. Just give it your best guess. I'll read each question and answer option and give you about 10 seconds before we launch the poll. Take that time to select your answer, and please note the letter corresponding to your selection. Once the poll launches, you'll only see the letter choices. We'll give you about 15 seconds to submit your answer. The answers will be discussed throughout the presentation today. So let's get started. Which of the following genomic features have the highest density of CpG sites? A, CpG island shores, B, enhancer regions, C, CpG islands, D, gene bodies. Note the letter of your selection, please. Question 2, which of the following tasks are best accomplished using supervised machine learning? A, discovering molecular substructure from a large cohort of neuroblastomas using cluster analysis. B, identifying the molecular subgroup of a tumor in a patient diagnosed with atypical teratoid rhabdoid tumor. C, determining if a tumor type of interest is a molecularly distinct entity by comparing it to reference tumors using T-SNE. D, all of the above. Note the letter of your selection. Thanks for the answers coming in. Question 3, which of the following metrics are used as a quality control for clinical methylation? A, CpG coverage and or mean detection p-value. B, median intensity values of methylated and unmethylated probes. C, probe level metrics. D, all of the above. Question 4, which feature of the Illumina Infinium 850K EPIC array make it well suited for the typical clinical workflow? A, works well on FFPE or frozen material. B, single base pair resolution across all available methylation sites in the genome. C, relies on methylation specific restriction enzyme digestion to fragment the genome. D, none of the above. Don't forget to note your answer letter. Question 5, in methylation base classification, what is the appropriate output when a tumor that is not in the original training set is encountered? A, the model should output a suprathreshold score for the control tissue. B, the model should output a suprathreshold score for the closest tumor class. C, the model should output a subthreshold unclassifiable score. D, the models are completely comprehensive, and do not encounter novel tumors outside the reference. Thank you for your answers. Question 6, which clinical decision or therapeutic strategy is most amenable to methylation profiling? A trial specifically targeting BRAF PV600E mutated tumor. B, a trial that reduces the radiotherapy for the Wnt molecular group of medulloblastoma and provides more intensive therapy for group 3 medulloblastoma. C, using the output of a methylation-based classifier to determine whether an ALK inhibitor is used on ALK fusion positive tumors. D, using methylation profiling to detect very subclonal MYC and amplification with the intent to escalate therapy. Today we are happy to have with us our presenter, Dr. Brent Orr. He's an associate member of the faculty of St. Jude Children's Research Hospital, where he is the medical director of anatomic pathology and the director of the anatomic pathology and neuropathology divisions. He has subspecialty expertise in neuropathology and particular focus on pediatric brain pathology. His high impact contributions to the literature in his fields of interest have led to inclusion as a member of the Consortium to Inform Molecular and Practical Approaches to CNS Tumor Taxonomy, or C-IMPACT now, and contributions to the WHO classification of tumors for both central nervous system and pediatric tumor volumes. Today, we look forward to his presentation on methylation profiling, recent findings, laboratory considerations, and clinical implications. Dr. Orr, thank you for joining us today, and feel free to begin when you're ready.
Okay, I want to thank the organizers, including Curtis Davies for inviting me, as well as Anthony for moderation. It's really a pleasure to be part of this seminar series, and I look forward to talking to you today. I have no disclosures. So the learning objectives today are to describe the basic biology of DNA methylation and the specific technologies used to detect DNA methylation in the clinical environment. I'm going to describe how tumor classification schemes can be refined using unsupervised methylation profiling, and then describe how methylation profiling can be used as a clinical diagnostic in the molecular pathology laboratory. First, a scientific overview. So I put this up as an example of the change in brain tumor diagnoses over time. And so if you look at the original diagnostic scheme back in 1927, published by Bailey and Cushing, there were only 13 classes. Now, the 2016 WHO has 155 classes. And the fifth edition, which we just recently published, has upwards of around 200 different classes. This reflects accumulation of cases, increased knowledge of biology, and advances in technology allowing for increased granularity of diagnosis. And I think if you overlay technologies on this plot, you'll see that you have an uptick in the 80s when IHC was introduced, then you have a little bit of a plateau. There's an uptick when next generation sequencing became more readily available. And I think what you'll see now is that as methylation profiling becomes more common, there will also be another significant jump in the type of and granularity of classification. So molecular methods are more precise, but they expand the classification schemes significantly. And so it used to be that we had histologically defined tumor types, including this example, medulloblastoma. With molecular methods, we've found that within these individual tumor types, there's actually multiple molecular subgroups, and they may cross boundaries for histopathologic types. These molecularly defined groups have different mutations, they have different transcriptional profiles, and oftentimes they have different clinical outcomes or responses to therapy. And so, it is an important determinant of patient outcomes. So if you look at classification of tumors in the modern era, there are benefits of increasing complexity of our classification scheme. So there's a better match of tumor type to therapy, homogeneity of individual groups improves the ability to elucidate biology, and we're better able to model and interpret data from individual tumor models. But there's also problems with increasing our complexity. So new entities are often rare, so it's very difficult for the average anatomic pathologist or neuropathologist, specifically in the community, to develop any experience with some of these rare entities. Clinical outcome data can't keep pace with the classification changes. And as far as the lab goes, increasing complexity of classification doesn't scale well in the clinical lab. So there's often a requirement for clinical validation of multiple biomarkers. So, you may need to identify the driver mutations in a tumor. You might need both a capture panel and RNA-seq or some other method to identify fusions. The other thing is that not all entities have specific mutations or genomic abnormalities that are readily detectable. So what I hope to convey today is that DNA methylation is a useful biomarker for the clinical lab. And so I think it's a powerful biomarker that fits well into standard clinical workflows. It is a universal biomarker that can be applied to even in tumors that lack defining gene mutations or fusions. It's especially a good method for molecular subtyping within individual tumor types. And the way that we implement it in the clinical lab relies on supervised machine learning models, which have potential to be augmented over time with added examples of tumors to existing classes or adding new classes to the model. And so I hope this is the take-home message, and then I'll talk you through why I think that these things are true. So I think it's good to take a step back and consider what is genetic versus epigenetic in the genome. And so genetics really refers to the heritable changes in DNA sequence or the number of genes or chromosomes. And that's, particularly under normal situations, most cells are getting the same general DNA sequence and the number of genes. Epigenetics is a heritable change in gene function that does not affect the DNA sequence. And so because all cells are getting the same basic genetic information, that isn't enough alone to specify the specific cells or tissue types. And that really is something that is left to epigenetics. The fact that they're both heritable make them both good biomarkers in the clinical laboratory if you can interrogate them appropriately. The two major components of epigenetics includes DNA methylation at CpG sites, CpG dinucleotides, and post-translational modification of histone proteins and chromatin. Now, of the two, DNA methylation at CpG sites appears to be the most stable and robust, harder to change of the two major epigenetic types, whereas post-translational modification of histone, it has a lot more lability and is a lot more dependent on the environmental stimuli. So epigenetics is a critical genomic regulator, and so some of the cellular processes regulated by DNA methylation specifically include gene transcription, cell type and tissue specification, genomic imprinting, chromatin structure, repression of repetitive elements, and innate immunity. And so, the innate immunity specifically is that when viruses insert into the DNA, they can be inactivated by methylation. If you look across the genome, the sort of major subdivision are repressed components of genome, which contain highly methylated DNA and methylated histones, and active components of genome, which include unmethylated DNA and acetylated histones. Now, again, I mentioned that these are heritable, so methylated, the signature of methylation is passed on to the daughter cells after replication, which makes it important way to, which we utilize in the clinical lab to identify specific tissue types. It's also important that that methylation signature also seems to be a blueprint for the way in which the chromatin is ordered in the next cell or after a dividing cell. So again, I think that these are important aspects in the way that we use it in the clinical lab. If you look at maintenance of DNA methylation in the genome, so this is the general way that it works. So you have unmethylated DNA, which is de novo methylated by the DNA methyltransferase 3 family of genes, and that's methylated on the 5', or excuse me, the 5th position of cytosine to form 5-methylcytosine. After replication, you have no methylation of the DNA after replication, so leaving you with a hemimethylated DNA. And so there's another form of methylation called maintenance methylation, which is modulated by DNA methyltransferase 1, and this is really the component that makes it heritable. So after you replicate, you're going to form a daughter cell, and that signature of methylation is then transferred to the daughter cell using DNA methyltransferase 1. Now, if there are ways in which methylation is removed in the genome, and so one is passive demethylation, and so if you have any sort of inhibition of DNA methyltransferase 1 after replication and are not able to copy those methylation marks over time of replication, you'll reduce the amount of methylation. There is also an active demethylation process in which TET proteins are used to oxidize methylation and eventually leads to base excision repair. This is really sort of outside the scope of this talk, but I just wanted to sort of mention that there is a both methylation and active demethylation process in cells. So if you look at DNA methylation in the genome, it's distributed unevenly, and so most of the genome is actually methylated and outside of specific regions in which remain unmethylated, and so particularly regions of heterochromatin have very high levels of methylation. CpG islands is a component of the genome that often has relatively little methylation, and so it has the highest density of CpG sites, but they tend to be hypomethylated. They're defined as greater than 500 base pair regions with greater than 55% GC content. And they're often in the setting of promoters, and so 40% of gene promoters contain CpG islands. There's also things defined as CpG shores, which are within 2 kilobases upstream or downstream of CpG islands. Methylation is also distributed in enhancer regions and some other regions throughout. As far as how DNA methylation regulates the genome, so in repetitive elements, there's often hypermethylation. In the normal state, this is repressive, and so in particular, transposable elements that may live in those regions are repressed. CpG island and CpG island shores under a situation of hypomethylation promote gene expression, whereas in hypermethylated state repress gene expression. CpG island shores also contain a lot of the tissue-specific differentially methylated regions and seem to correlate best of all the sites with gene expression. Enhancer regions can be positively or negatively regulated by methylation, and it largely has to do with which proteins may be binding in those regions. Some of these other regions do have methylation, but are a little less characterized. So there is dysregulation of DNA methylation oftentimes in cancer, and certainly as molecular pathologists, they have an understanding of that. So in those repetitive elements, hypomethylation leads to genomic instability and re-expression of transposable elements. In the setting of CpG islands or CpG island shores, hypomethylation can lead to expression of oncogenes, whereas hypermethylation can lead to repression of tumor suppressors or DNA repair enzymes. And so we see this sometimes in the clinical lab for methylation of RB1 or DNA repair enzymes such as MGMTs. There can also be dysregulated gene expression depending on the methylation state in enhancers. So if you look at the two main types of DNA methylation in the genome, the major is CpG methylation, and these are sites, this is the most common, so this is a CpG dinucleotide, and that makes up about 99.8% of the methylation in the genome. You can see that here. There is also some non-CpG methylation. Just wanted to make this group aware. Those would be CA, CT, and CC sites. It represents a very small fraction of total, particularly in differentiated cells, and may play a bigger role in embryo. So we detect DNA methylation experimentally using bisulfite conversion. So the way that works is after bisulfite conversion, unmethylated cytosine is converted to uracil, whereas methylated cytosine is protected from conversion and remains as cytosine. After you perform amplification, either PCR or non-PCR, unmethylated cytosine is converted to thymine, and then methylated cytosine is retained as cytosine after amplification. And so really that difference in C or T in the amplified product helps one determine whether the original DNA was methylated or not. There's several different methods that can be used to detect methylation. And so here's just some examples. One, reduced representation bisulfite sequencing uses enzymatic digestion and can cover about 85% of CpG islands for about $300. The most common method, particularly in the clinical environment, is methylation arrays, often the Illumina array from EPIC array from Illumina. That has 850,000 sites, represents 99% of RefSeq promoters and 95% of all CpG islands. The general cost of that is about $350 to $450 per sample. Illumina has also put out a TrueSeq capture sequencing panel that can capture 3.3 million CpGs and has comparable target representation to the EPIC array. It's a little bit more costly and not as amenable to working in FFPE. The gold standard for methylation detection would be whole genome bisulfite sequencing, which gives you base pair resolution, but is quite costly and not at this time utilized in the clinic very often. So looking at this Illumina array, which is the one that we typically use in the clinic, the general process for this array is that the DNA is bisulfite converted. There's a fragmentation and whole genome amplification. There's hybridization to the bead chip, and where the array contains two different types of beads. One is type 1, which contains methylated and unmethylated beads at each genomic loci. A type 2 bead has allele-specific annealing with single base extension, and so the extension is only possible when an adjacent base matches, and then the signal provides the signal provided by the tags on the extension bases tell you whether the site is methylated or not. These are read with an iScan instrument, which is a whole separate instrument. The newer instruments, I think there is also ways in which the NextSeq, particularly 550 instrument, can be used to read the arrays. In our clinical lab, we have a separate instrument for sequencing and iScan. So the data that you get back, the array data is stored as a beta matrix at each CpG site. And so from the 850K array, you get a beta matrix, which is a high dimensional matrix representing methylation state at each CpG site on a scale of 0 to 1. So it's basically a ratio of methylation at that specific site across the bulk sample. And if you run all the CpGs from an individual sample out, you can see this typical two peaks. And so one on the peak on the left-hand side represents hypomethylated DNA, whereas the peak on the other side represents hypermethylated DNA. And the typical sample will have different proportions, but a peak of each of these. here's our first question. So which of the following genomic features have the highest density of CPG sites? Is it A, CPG island shores, B, enhancer regions, C, CPG islands, D, gene bodies? Okay, yes, the answer is CPG islands. It looks like most everyone got that one correct. So, we've talked about a little bit about the basics of DNA methylation in the genome and how we detect it, but you know, really the use of this as a tumor diagnostic really started, I think, at least in neuropathology, with this paper coming out of a Heidelberg group. So I mentioned that you know, we knew of 4 molecular subgroups of medulloblastoma, and those had been defined based off of transcriptional profiling. And so the importance of this paper is they use these high-density methylation arrays to demonstrate that those transcriptional groups that we knew about, the Wnt, hedgehog, group 3, and group 4, could be recapitulated using the methylation arrays. And so the way that they did this is they ran all the tumors on the arrays. They looked at the differentially methylated regions, and they did perform cluster analysis or dimensionality reduction. And so this is all unsupervised analysis, just looking at the standard deviation of the probes. All the colors have been overlaid after demonstrating that it really does fall into these 4 groups. So it turns out that this is not just a feature of medulloblastoma. So this came from a paper we published in 2016. And this is a reference series of lots of different tumors we were comparing to PNET at the time. But it seems to be a normal feature of tumors that that specific molecular types of tumors group together by their methylation state. And so you can see here, either by cluster analysis or TSNE, which is a dimensionality reduction method, you can see specific molecular groups of high-grade gliomas falling out, specific ependymoma groups falling out, medulloblastomas falling out into distinct regions. I'm going to show several of these TSNE plots, but what this basically is, it's taking the high dimensionality data, in this case differentially methylated probes, and it's projecting it down on a 2D space. And so each dot is a tumor, and if tumors are close together, that represents a higher or a closeness in high dimensionality space as well. And so distinct tumor types are associated with stereotypic methylation signatures that is reproducible, and we think that this represents some combination of cell of origin and specific mutational driver. And so I think it's important to distinguish between the 2 ways in which methylation data are used, both in the literature and in the clinic. And so one is using unsupervised methods. In unsupervised methylation analysis, the data is unlabeled on analysis. So we're usually just using something like standard deviation across all the probes in a cohort to look at the differentially methylated regions. You know, computational methods are used to derive insight on the molecular structure of the data, including cluster analysis or dimensionality reduction. The common ones you'll see is TSNE or UMAP. And often after visualization, you can see a lot of overlay of some sort of molecular attribute or some sort of diagnostic attribute. And so that's unsupervised method. Now the clinical implementation of methylation profiling is a little bit different. And so there we're using supervised methods. And so in those, we get reference classes of labeled data, so representative tumors from specific classes we want to detect. We build machine learning models to train on the labeled data to predict new classes that we might encounter. And the output is a class score probability of class assignment. So in the little schematic here, we've got 3 different classes, red, blue, and green. We train a model, and then when we encountered a new unlabeled sample, we get some sort of class prediction, and in this case, it gets a low score for everything except for red. And so we would predict that the new unlabeled sample is the red class. Okay, going back to unsupervised analysis and how it's been used. I think you've probably seen a lot of papers out there that define molecular substructure in some sort of large histopathologic tumor type. And so, so what often happens is you get a cohort together, you run unsupervised analysis, and you see this substructure falling out. And so in this case, it's forming 3 different discrete subtypes. You run orthogonal validation, so NGS, directed sequencing, FISH, copy number profiling, a lot of other things, compare it to clinical outcome data to make meaning out of these clusters. Sometimes people use this to uncover new molecular subtypes. So you may be a tumor type where you have 3 known molecular subtypes. You add additional samples and perform unsupervised analysis, and you see splitting of those types into 2 subtypes. So in this case, 3A, 3B. And again, all of this is done with orthogonal validation, with sequencing and molecular and other molecular and clinical data. And so there's been a huge expansion in the brain tumor realm for defined molecular subgroups based on unsupervised analysis of DNA methylation data. And so in the new WHO, which was just recently released, there's 13 molecular groups of medulloblastoma now. So the original 4 groups have been subdivided even further, and importantly, there seems to be different risk, and so we can pull out subtypes that have lower risk even out of what in the group 3, group 4, which are traditionally higher risk groups. There's 10 molecular groups of ependymoma that have been described in the new WHO, 3 groups of ATRT, and greater than 6 high-grade gliomas. And so all of that data has really come from unsupervised analysis of methylation, or all these groups have been born out of unsupervised methylation analysis. We also use unsupervised analysis to resolve unclear diagnostic entities. And so the way that that works is you take a reference cohort, and in this case, say reference 1, 2, and 3 in the schematic, of tumors that you know what they are, and you have good mutational data, copy number data, and transcriptional data to suggest that they fall into a specific molecular group. And then you add test samples, oftentimes from some ambiguous diagnostic entity, and then you perform cluster analysis, and again, lots of orthogonal testing. And so this is really, so it really often find one of 3 patterns when you do this sort of analysis. And so you see the test samples that you've added fall into a specific known molecular entity, and we would interpret that as a known molecular entity. Sometimes you see that the test samples sort of fall into multiple groups, and we interpret that as it's a heterogeneous molecular entity, so that the diagnostic samples do not represent a single molecular group. And sometimes we find that the diagnostic samples or the cohort that you put into this form a novel molecular entity. And when you follow that up with sequencing, often those are defined by specific molecular drivers. And so my lab has been involved in several studies that have really used this last approach to try to resolve some unclear diagnoses. And so it started with gliomatosis cerebri back in 2016, which was always sort of a tenuous diagnosis in the neuropath community. We showed that it was really not a single molecular entity, but really heterogeneous presentation from lots of different tumor types, or high-grade glioma tumor types in particular. And so it led to its removal in 2016 from the WHO. We followed that up with looking at CNS PNET, which again was a diagnostic challenge in clinical neuropathology, because it often was being the distinction between high-grade glioma and PNET was often distinction. And if you called something a high-grade glioma, it led to local radiation, whereas if you call it a PNET, they would get craniospinal radiation, which is quite a big difference. We found that CNS PNET was also a heterogeneous entity. It led to its removal from the WHO. Now, in that paper, we also found that, and this is a summary of published works, so I didn't want to present a lot of the basic findings, but in that PNET paper, we also found the power of methylation profiling to discover novel brain tumor entities. And so we discovered 4 different brain tumor entities with specific drivers in that paper, and they've been added to the updated 2021 WHO. There's been several others, an additional subtype of diffuse midline glioma, which has been added to the updated WHO, and we've also been involved in pediatric adrenocortical tumors, which we find falls into 2 molecular groups. We find that radiation-induced gliomas are a specific molecular group or fall into a specific molecular group, and we've also identified novel groups of molecular groups of rhabdomyosarcoma. Okay. This is just, I put this slide in here. These are the 4 molecularly defined brain tumor entities that we described. One, CNS neuroblastoma with FOXR2, one, Ewing's family of tumor with CIC, something we called high-grade neuroepithelial tumor with B-core ITD, and then something we described as high-grade neuroepithelial tumor with MN1 alteration. Some of the names have changed in the new WHO, but when you go in and sequence, so these very defined molecular groups from the methylation profiling, on sequencing, they're often defined by specific fusions or genomic drivers. And so we found FOXR2 abnormalities in the one, the CIC tumors in the brain had a high proportion of CIC NUT-M1 fusions, and they seem to be somewhat close to the CIC DUX4 fused tumors in the periphery. There's a B-core ITD in exon 15, and these was now being called CNS B-core altered or CNS tumor with B-core ITD in the new WHO. It's similar to the abnormalities that are found in others tumors outside the brain, including clear cell sarcoma of kidney. And then the final one was this MN1 altered tumor, which will be called astroblastoma NM1 altered, which had a high proportion of CNS of MN1 Ben2 fusions. So again, the methylation profiling was the starting point, but then we're also identifying these drivers by sequencing things within a specific cluster. And these are all quite rare tumors, and so it is a nice way to identify novel tumor groups. So this unsupervised analysis can also have benefit to conventional test development and validation. And so, after we knew of these tumors and that they occur sometimes in children, we were able to bring up conventional testing to help diagnose these in the clinic. And again, it took multiple validations of multiple different markers, but we were able to develop NUT-M1 immunohistochemistry, CIC FISH to recognize that group, B-core immunohistochemistry, and a PCR for the B-core ITD to recognize that group, MN1 FISH. Now, the CNS neuroblastoma group is quite hard to detect outside of methylation because of the way in which the FOXR2 abnormalities are formed, which include deletion of intervening space between closely approximated genes. Okay. So far, I've only talked about unsupervised analysis of methylation data, but supervised classifiers are critical for clinical implementation of methylation profiling. And this is a paper which I think probably a lot of people are familiar with. They came out in 2018 from the Heidelberg group. And what they did in this, and I think this was really a landmark for where the field is headed, is they took a reference series for all brain tumor types that were in the 2016 WHO. It includes 75 tumor families, 91 total subclasses. 7 of those subclasses include control tissue in the brain. They made a reference group. So each dot on here, this is a similar T-SNE to what I showed previously. Each dot represents a tumor, and so they had representation from multiple instances of the specific tumor types. And their total reference series was 2,801 tumors. They trained a random forest classifier, which is a machine learning model to do supervised classification, and they output a probability score for family and class. They very wisely built a externally facing website that you can upload IDAT files and output a molecular classification of the tumors. Okay. So digging a little bit deeper in the basic steps in supervised classification, which again is should be distinguished from unsupervised analysis. So these are the basic steps. So first is the reference cohort. So examples of samples in the specific methylation classes to predict, labeled by class. Then you do a probe selection. So and what happens in probe selection, you're reducing the probes to the specific probes most useful for discriminating methylation classes. So, the common platform right now is the 850K array, which is 850,000 sites across the genome. The actual molecular classifiers only use about 50,000 of those sites to distinguish all 91 classes. Then is the supervised classification model. They used a random forest classifier, but there are a lot of different options out there as far as supervised classification models. And these are machine learning methods that use selected probes to predict the class labels. So the supervised model outputs a score for each reference class, with the highest score representing the model prediction. Now, an important part for the clinical implementation of this was the calibration model at the end. And so when you train these machine learning models, the supervised classification models, the distribution of the class level scores is slightly different. And so that calibration model is important to place the output of the supervised model on a uniform scale. So it allows you to have uniform clinical thresholding. So in their implementation, they use 0.9 as a class score of their calibrated class score, as their threshold, and that is also something that we use clinically. And so when you implement these in the clinic, after you've trained this model in this manner, you encounter a test sample which doesn't come with a label, right? It could be coming from the clinical service or a research test sample. You use the trained model to take the input from the specific methylation sites, from the selected methylation sites, maybe 50,000 probes, and it outputs a raw score for each class. And the top class score is the prediction, is calibrated, and you can do clinical thresholding reporting. Now, what's nice about this system is that it's comparable over time, and so it becomes an institutional memory of specific tumors. And so even rare tumors, if you collect them over time, you can use those new samples to improve the model. And so by either adding examples to existing classes or if you identified new molecular subtypes of tumors or new tumor types, you can add those into your training set. Okay, move on to question 2. Which of the following tasks are best accomplished using supervised machine learning? So A, discovering molecular substructure from a large cohort of neuroblastomas using cluster analysis. B, identifying the molecular subgroup of a tumor in a patient diagnosed with atypical teratoid rhabdoid tumor. C, determining if a tumor type of interest is a molecularly distinct entity by comparing it to reference tumors using t-SNE. Or D, all of the above. All right. So sorry. So it looks like, so the answer is B, identifying molecular subgroups of tumors in patients diagnosed with atypical teratoid rhabdoid tumor. So the other two were actually unsupervised methods. So cluster analysis is an unsupervised method. T-SNE is a dimensionality reduction, which is also unsupervised. And so this is determining the molecular subgroup in the clinical specimen relies on supervised classification. So now I'm going to move on to laboratory considerations. So this is a general workflow for DNA methylation array profiling. So we're often using FFPE material. We extract the material using a Maxwell, a very standard process in the molecular lab. Usually requires about 150 to 350 nanograms of DNA. We're running on the A50K array, which is a 5-day workflow. You really cannot reduce it less than 4 or 5 days. We're running about 1 batch a week. There's 8 samples per array, and so without batching, you sort of waste a lot of reagents. And then after we read it on the iScan and then put it through the bioinformatic pipeline, the 2 things that we're reporting are methylation class and copy number abnormalities. So one of the considerations in the clinical environment is which algorithm to use for supervised classification models. And so the original model used reference series and then trained on a random forest. We ended up at St. Jude implementing a neural net model, and we validated that using cross-validation, which is partitioning out the training set in different partitions to get an idea of how well the model performs on each class. And then we've validated that with 2 different sets. One was a publicly available set of about 1,000 tumors, 1,100 tumors with 450K data, and then a second St. Jude validation set with close to 1,850K data samples. We picked the neural net model because in our hands it showed higher proportion of samples with classifiable scores without degradation of accuracy compared to the random forest. And so I don't really have time to go into the data here, but you know, the model performance was almost identical, maybe a slight edge in our neural net compared to the original random forest described. But the biggest difference was that our model was more confident in its scores, and so we didn't have as many ambiguous samples with low threshold scores, below threshold scores. As I mentioned, we are also reporting copy number data, and it can be extracted from the DNA methylation array. Essentially, it uses the array almost like a SNP array. There's an algorithm called ConyMe in the R package, which is an R package that can be used to extract copy number out of the array. This is just some example data using IGV view of the segmentation files, and so you can see, you know, ATRTs are showing that very common loss of chromosome 22. Wet medulloblastomas are characterized by a high proportion of monosomy 6, which you can detect pretty readily out of this array. Group 3 medulloblastomas often have amplification of MYC, and we can detect most of the common oncogene amplifications using the array. And you can see that focal MYC here, and if you zoom in, excuse me, many of the group 3 medullas are showing MYC amplification. Okay. So the array profiling is amenable to standard pathology laboratory workflows, and so this is a dilution series from the original paper demonstrating that you see good correlation between the methylation values, even diluted out to 100 nanograms. After that, you start to see a drop-off. We've recapitulated this in our lab, and so we usually ask for between 125 and 350 nanograms of DNA. It also, an important aspect, particularly implementing in a clinical environment for solid tumors, is that it works comparably well in FFP and frozen tissue, and so we don't see much drop-off when comparing classification scores on FFP or frozen tissue. And this is just comparing a sample for FFP and frozen by the methylation values, and you see good correlation, R of 0.98 between those. So Our clinically live assay, we've had close to 600 cases. Most of that is coming from FFPE slides, and so 492 of the 590. We've obviously had some scrolls, but also a little bit of fresh DNA. All of them work pretty well as long as you can enrich for our tumor content. I plotted the total DNA that we got by the number of slides, and you can see that 95% of the cases submitted had sufficient material for testing. Our range was 0 to 9 slides, but the median was around 3, and you can see pathologists are basically ordering 3 to 5 slides on most cases. And again, we're asking for enrichment for greater than 70% tumor. So we found that there's a drop-off in the classification ability if you lower too much below 70% tumor. The quality control metrics that we're using in the lab. So we use 3 main quality controls. So the probe mean detection p-value of the array, and so this is again derived from an R package called Minify. We use a cutoff of 0.05 or 95% of CPG sites detect at a threshold of 0.05. You can plot that 2 different ways, but the way that here's some just example data. So on the left, the low values are good, and the failed sample has a higher than 0.05 mean detection p-value. If you look at the CPG coverage from 0 to 100, again, the low sample, the blue here. And that seems, this quality control seems to correlate very well with whether this sample will behave well in the clinical lab. We also evaluate median intensity values of unmethylated and methylated probes. And so we plot it as a plot like this, a scatter plot. And what you want to see is that the samples are above 12 on both of those metrics. At least that's ideal. So the more that those are up in the top right corner of your plot, the better. When samples fail, they tend to be in this bottom left-hand corner with low intensity values. We also use EWAS tools package in R to look at 17 probe level metrics. So I've just listed them here with our cutoffs. We find that these are not always the best predictor of whether a sample will fail. The bisulfite conversion for the green and red probes tends to be the most predictive. Some of these others are a little bit variable. The output of copy number analysis can be used for quality control as well. And so this is sort of an unofficial quality control, but on the left, I'm showing a control sample with no QC failure. You see this very tight copy number profile. On the right, I'm showing a sample that has had failed bisulfite conversion. And so you see this dispersion of the copy number profile after the Kanye algorithm. Okay, question number 3. Which of the following metrics are used as quality control for clinical methylation? A, CPG coverage and or mean detection p-value. B, median intensity values of methylated and unmethylated probes. C, probe level metrics, or D, all of the above. Okay, looks like the answer is all of the above. It looks like everybody got that, or most everybody. Question number 4. Which feature of the Illumina Infinium 850K EPIC array make it well suited for the typical clinical workflow? A, works well on FFPE or frozen material. B, single base pair resolution across all available methylation sites in the genome. C, relies on methylation-specific restriction enzyme digestion to fragment the genome. Or D, none of the above. All right, go ahead and put in your answer. All right, the correct answer is A, works well on FFPE or frozen material. This final section of the talk is to talk about the clinical implications. And so I talked about some of the features of the details being in the clinic, but how does it matter? What does it matter? And so we implemented methylation profiling at St. Jude October 2020. Again, we're reporting methylation class and copy number data. We've run close to 600 samples to date, and it was introduced only in the neuropathology service. Our turnaround time is 1 to 2 weeks. Again, that really has to do with the 5-day workflow and the need to batch samples. So we run about a single batch run a week with 1 to 4 arrays. I've collected follow-up data on 515 of the cases, and I'll present some of that data here. So the top of the screen will be absolute values, and the bottom will be % of total samples out of 515. And so one of the questions was how many of the samples are we hitting the clinical threshold of 0.9, which is our cutoff. And it looks like a pretty high proportion, so about 85% of the samples are getting a classifiable score above clinical threshold. That matched pretty good to our validation data. If you look at the sub-threshold samples and break those down by whether we expected them to be in the class in the reference or not. So 18 of those were likely correct samples out of 77, meaning that we believe that they probably were classifying correctly. They were just sub-threshold scores. 59 of those out of 77 were tumors that were sub-threshold and likely were not part of the, or were truly unclassifiable. And that comes out to about 11%, which is pretty similar to what the German experience was with this specific reference series. If you dig a little deeper into those unclassifiable tumors, 37 of those were not in the reference, 10 were unclear, 8 were expected in the reference, but received very low scores, suggesting that they might not be, and a couple were suboptimal specimens in which there seemed to be some mix of tumor and normal, which made it unclassifiable. The typical thing to happen is that if you have normal brain contaminant, that it gets classified as a normal control class, but there is some sort of middle of the road where you have some enough normal mixed with the specific tumor type, which can cause unclassifiable score. If you look at the tumors that were above threshold, 426 out of 438 appeared to be concordant with the clinical diagnosis, meaning that they seem to be reasonable classifications. That's about 83% of the total cohort. 2% or 12 samples were what we believe to be misleading classes. If you dig into that a little bit more, out of those 12, 4 were low-grade tumors that had what we thought were a reasonable amount of tumor content and still got classified as control tissue. There were 4 sort of minor misclassifications, and those were tumors that were believed clinically to be some specific type of low-grade neuroepithelial tumor, either low-grade astrocytoma or low-grade glioneuronal tumor, and it got classified as another, and we had good evidence that it probably was not correct. And 4 were sort of considered major diagnostic errors, and that it would change therapy, and so those would be things like typically astrocytomas that were called some low-grade astrocytoma group, but actually classified by the classifier, but histologically were quite aggressive. Now, one of the things that we're interested in is how much added information was added to these. And I think that this is pretty preliminary because it was a little hard to abstract out whether people were waiting or how often people were waiting to sign the case until the classifier had resulted. But in 399 out of 426, it confirmed a clinical diagnosis. A proportion of tumors, it clarified an ambiguous diagnosis of something that was very difficult histologically, and it suggested a tumor type that helped clarify. In 4 cases, it suggested an alternative diagnosis that we could later prove that was correct by some other molecular means. And the important thing, in a very high proportion of cases, almost 45%, we added some sort of molecular subtype that couldn't readily be ascertained in the clinical environment. So this is a way we're getting about 45% of our cases. We're getting molecular subtyping within specific tumor types using a single biomarker. I've got a few example cases to show right before I close out. And so here's an example case where we've extended the clinical subtyping. And so at St. Jude, we run an IHC on medulloblastomas to molecularly subtype things into Wnt hedgehog or non-Wnt non-hedgehog. In this case, I diagnosed this as a medulloblastoma, large cell anaplastic, non-Wnt non-hedgehog with MYC amplification. Our IHC stain cannot separate things into group 3 or group 4, and the group 3s tend to do poorly. Group 4s do a little bit more intermediate. After methylation profiling, we get this nice copy number chain profile demonstrating isochromosome 17q, as well as a classifying score of 0.93 for subclass. And so we've extended the ability to subtype in this instance. Here is another example. So there's not a good alternative biomarker for a specific tumor type. And so in this case, it's a 16-year-old female with a spinal tumor. The outside diagnosis was pylomyxoid astrocytoma. Some of those would have BRAF fusions, but not all. My diagnosis was myxopapillary ependymoma. Myxopapillary ependymomas have some histologic features that are you can use, but there aren't a lot of good molecular means to sort of prove that you're correct. After methylation profiling, it received a very high score for 0.94 for myxopapillary ependymoma. Now, being a children's cancer hospital, I don't see a lot of 75-year-olds, but here's another case that was sent. This was one that I accepted. It was a 75-year-old male with a history of choroid plexus tumor 15 years prior to presentation. He presented with a new mass in the spine, and so the outside diagnosis was metastatic prostate cancer. The consulting pathologist called and said, I wonder if this could be a late metastasis of the original choroid plexus tumor. And the histology was quite fried here, making it a little bit hard to identify what the tumor type was on conventional testing. We ran it on the classifier and received a very strong score for some adult type choroid plexus tumor. So it's a nice way to resolve this clinical problem. And here's the last one. So in non-classifiable tumors. And so this was a 13-year-old male with reported NF1 and previous pilocytic astrocytoma, now with a new tumor in the radiation field. And so my diagnosis was, it was quite a difficult tumor, and unfortunately I don't have a picture of that, but a malignant tumor with rhabdomyoblastic differentiation. When we ran it on the classifier, it had a very deranged copy number profile and no methylation class above a reportable threshold. So I think as high as class score was 0.03. Later, after sequencing, because the patient became a patient in our lab or in our institution, we found dicer1 frameshift mutations, and so the updated diagnosis was primary intracranial sarcoma, dicer1 mutant. And so, this is just a good example of what happens if you have tumors that are outside the reference classes where they get no score. Question number 5, is methylation-based classification, or in methylation-based classification, what is the appropriate output when a tumor is not in the original training set? A, the model should output a suprathreshold score for control tissue. The model should output a suprathreshold score for the closest tumor class. C, the model should output a subthreshold unclassifiable score. And D, the models are completely comprehensive and do not encounter novel tumors outside the reference. So go ahead and put your scores in. Right, so it looks like most people got it correct. The model should output a subthreshold unclassifiable score. Okay. Last question. Which clinical decision or therapeutic strategy is most amenable to methylation profiling? So A, a trial specifically targeting BRAF V600E mutated tumors. B, a trial that reduces the radiotherapy for the Wnt molecular group of medulloblastoma and provides more intensive therapy for group 3 medulloblastoma, so that's a subtyping task. C, using the output of methylation-based classifiers to determine whether an ALK inhibitor is used on ALK fusion-positive tumors. So that's a targeted inhibitor. Or D, using methylation profiling to detect very subclonal MYC amplification with the intent to escalate therapy. So go ahead and put in your answers. Right, so the answer in this case is a trial that reduces radiotherapy for Wnt tumors and provides more intensity for group 3 medulloblastomas, which again is a subtyping test. These others are things that, so A and C are things that really are probably more well suited for sequencing-based approaches. D is subclonal amplifications are not well detected in this assay, and so that's better. FISH is a better way to detect very subclonal copy number abnormalities because this is a bulk method. Okay. So here's my summary. So I hope that I've been able to convince you that DNA methylation profiling is a powerful biomarker that fits well into standard clinical workflows. We're high on this because we think it's universally applied, even in tumors that lack defining gene mutations or fusions. And it's especially good at molecular subtyping within individual tumor types. And again, this ability to update your models over time. So it uses supervised machine learning models, which have potential to be augmented over time, which we think is a really nice approach. Here are my acknowledgements. There's obviously I didn't do all this myself, and so just quickly, Alex Brewer, Stan Pounds, and Tong Lin did a lot regarding the informatics in our clinical implementation. Larissa Furtado was instrumental in getting it validated in the clinical lab. Ruth Tadavosian, Surya Allen, and others did an excellent job helping us get validation samples together. And I'm happy to take any questions.
Thank you, Brent, for this excellent presentation. We've received a few questions from the audience that we can we'll present to you and you can address. I really appreciate this this presentation. It was great. A couple of us were wondering about specimens who might sort of fail the 70% threshold. I had a question about that. So you touched on a lot of this data. Did that data that you ran include samples that were less than the 70% threshold that you tried, or were they all over that threshold? And then I was a little curious about how you assess that threshold. Is that 100% sort of eyeballing it by the pathologist, or do you do any correlation with allelic fraction from sequencing or anything like that?
Yeah, it's a bit of a mix. And so, yes, the bulk of it is done, is actually by eyeballing it by the pathologist, which I suspect in this crowd there's some skepticism. But yeah, so that would be more like 70% based off of the pathologist's assessment, probably more accurate. And it's not, just to speak to that, it's not that you won't get anything below 70% that classifies correctly. The issue is that the variability is higher. So you may get less tumors falling into a classifiable score. Oftentimes what happens is you end up pushing it towards a control class, or you get a sub-threshold score. So 70% is, really what we're saying for optimal to get tumors that have classifiable scores and are correct.
Great, thanks. All right, I have another couple questions. Since we're over, we can just take a couple of these and then we can head out. One of the questions first on the list was for the unsupervised analysis, do you have to have an a priori knowledge of what the comparator tumors should be?
Not exactly. And so, I mean, I think it's helpful in some ways to make sense of the data, but I don't, if you took all comers of tumors and did unsupervised analysis, you would see some structure falling out. It's really just a matter of making sense of the data. It's sometimes useful to know what you're dealing with up front, if that makes sense.
A couple of people have asked about differences between pediatric versus non-pediatric tumors. Have you seen any difference in pattern or used any different algorithms for this analysis between groups?
So the reference that we use has representation from pediatric and adults. We have, because the German group was, trying to make that pretty comprehensive for brain tumors. We have some indication from several different tumor groups that the pediatric representation of specific tumors is slightly different than adults. And so we did a study with Michael Clay, who's now in Colorado, where we looked at pediatric adrenocortical tumors. There was a large cohort of ACTs in the adult TCGA data set, and it looks like the ones that we find in the pediatric population show different molecular substructure than those in the adults. It's true of oligodendrogliomas in adults and peds, and so tumors that show oligodendroglioma morphology in the pediatric population often have FGFR1 abnormalities and cluster closer to dysembryoplastic neuroepithelial tumor, whereas the adult type have 1p19q deletion and IDH mutations. And so I think that they're definitely, we also saw in rhabdos, so the adult type rhabdos, which tend to be these pleomorphic rhabdomyosarcomas, group different, showed different molecular group than the embryonal and alveolar. So, I think there's a little bit of overlap in that some of the what we would call adult type tumors sometimes are presenting in kids that are post-puberty or things like that. But oftentimes what we're finding is that there's distinct adult and pediatric manifestations from a molecular standpoint.
Great. Maybe one more, since we're kind of running over a little bit. Someone's asking if you've attempted to perform the clinical assay with any circulating tumor DNA or any liquid biopsy applications.
We haven't, and I think that actually is a really interesting question or approach, and certainly I think that probably it's certainly something we've thought of, but I think it's the implementation of that is quite difficult. And I'm not 100% sure that the probes that we use on the clinical classifier would be the exact probes that you would want to use for that approach. I guess is the short answer to that. I think you would maybe have to do a different probe selection or potentially just build different classifiers. But you know, theoretically, it would be a really interesting way to go about it.
All right. Well, thank you so much, Dr. Orr. On behalf of AMP, I'd like to thank you again for providing us with your time and expertise on the current state of methylation profiling. This was a great talk. We'd also like to thank the audience for attending and for your thoughtful questions. We're pleased to announce that this webinar is eligible for free continuing education credit. The instructions for how to claim CME or CMLE credit will be sent to you via email shortly. Please be aware that you cannot claim both CME and CMLEs for the same content. AMP Education will send a follow-up survey to attendees in the near future. The valuable feedback that you provide will help us improve our offerings so that you get the kind of content that you need and want. We will use this anonymized information to design new content and to keep costs for webinars low or free, as in this case, for everyone. This webinar was part 8 of our exciting series titled Emerging and Evolving Biomarkers, Recent Findings, Laboratory Considerations, and Clinical Implications. The next webinar in this series will be presented by Dr. Reina Shan on immunoglobulin and T-cell receptor gene rearrangement sequencing, and will take place on Thursday, April 7th, 2022 at 11 a.m. Eastern. Register now at amp.org/EEB or check out the full collection from this series in that same section of the website. Finally, we'd like to let you know about another free series that might be of interest to you. There's a series called Utility of Cell-Free DNA in the Clinic, Current State and Future Directions. It's an 8-part webinar series that covers a range of concepts from the basic biological underpinnings of cell-free DNA to new and specific advances in the diagnostic and prognostic use of cell-free DNA and circulating tumor DNA. The 5th webinar in this series will be held on March 24th, 2022. You can register now or get more information about these upcoming webinars by going to amp.org/LQB. On behalf of AMP, the EEB Content Committee, and myself, I thank you very much for attending today, and this concludes our presentation.